Good afternoon, and thank you again to Ellipse for this invitation. Um, so I'm actually here on behalf of this company in Gurgaon called Prodomics Technologies Private Limited. We're about 4 years old, and the major focus ever since our inception has been in the field of mass spectrometry and particularly metabolomics for multiple reasons. Because if you want to actually get insights into human health, the metabolome or the composition of metabolites provide you the direct reporter system for health states, in contrast to genes, proteins, whatever. Yeah, because metabolites are end products of body biochemical reactions. Uh, so they, in a sense, they give you a snapshot of the biochemical equilibrium that's established in the body, and that was at least was the working hypothesis we start with. And any perturbation in the health state or the health equilibrium is driven and perpetuated by alterations in or perturbations in biochemical reactions. So therefore, if you can actually capture the metabolome, which is the complete spectrum of metabolites in a biological fluid, you should be able to track what's happening to health. And health is a dynamic process, and metabolomics or the metabolite composition tracks that dynamism in changes in health. Because genetic mutations are static, they do not tell you changes as a function of time, whereas metabolomes do. Yeah. So we've been focusing on cancer, and the obvious thing about cancer is, I mean, obviously it is a disease of importance. I won't. Uh, and the core issue in cancer is that when you have, because the high mortality rate that you see is primarily because you detect it too late. On the other hand, if you can catch it early, say in stage 0 or earlier, uh, you certainly improve the prognosis, and at least in terms of numbers, you can actually reduce mortality by as much as 70% or so, right? And that is, but the trick is the early stages of cancer are asymptomatic, and how do you actually catch them, right? And this just shows you how you can improve prognosis and 5-year survivals if you can catch some of these cancers early. And in general, the ways, the various methodologies that you have for screening or detection usually involve medical procedures, or protein biomarkers like you have in prostate, PSA, and so on, or medical imaging. And there are, of course, serious issues with all of this, particularly in terms of the accuracy and so on. And more importantly, these are a little more cumbersome, particularly for early stage cancer detection. And that's been our focus all along, is early stage cancer detection. So when we started off, we wanted to address the question of what we thought was needed is a technology that detected cancers early, and obviously it had to be non-invasive. Specificity is clearly important since we're thinking of a screening tool, and of course economical, and one way to achieve that is to integrate multi-cancer detection into a single test. Yeah. So this is something there is a global recognition of, and there are companies and academic labs looking into it. And a lot of these efforts are actually focused on looking at using this technology called cell-free DNA. And I'm also including circulating tumor cells in this. One of the problems with that is, the problem is when you talk about early stage cancers, there are very few circulating tumor cells, and also very little of circulating tumor or cell-free tumor DNA that is in circulation, right? And so there is a serious quantitative limitation that comes in there. So one problem is you have to collect a large amount of blood, number one, and there also the best sensitivity you can actually achieve across the board is not more than 30 to 40% when we're talking about early stage cancer detection. Our hypothesis was, when you actually look at it biochemically, cancer is fundamentally a metabolic disease, right? The cancer cell is continuously dividing, and in order to achieve that, it has to ramp up its metabolism. I'm sure you've all heard of things like Warburg effect and so on. And therefore, if you can actually capture signals that define these altered metabolic states and so on, you may have a more accurate handle on capturing cancer. And I'll show you that actually turned out to be successful. Um, yeah, so that's just simply shows the divide cell. Just summarizing the endpoint here. So for our technology, you hardly need very little blood. It's a serum-based method, but you hardly need about zero point 0.1 ml of serum for this test, and we can achieve early stage detection accuracies, as I'll show you, of greater than 98%, and specificities too of greater than 98%. Um, So what is the logic here? Like I said, you're looking at changes in metabolism and through a readout on the metabolite patterns that exist, in our case, in serum. And I'm just illustrating this with an, this is some of our own earlier studies, but not to worry about it. What this shows metabolic pathways. This is actually glycolysis, citric acid cycle. This is a core metabolism that a cancer cell, this must be affected in order for a cancer cell to continuously divide. But the point is, you can actually measure each of these metabolites in each of these states, whether glucose 6-phosphate, fructose phosphate, and so on, and you can measure these. And we did, and if you do things like if any particular reaction is perturbed, all the other metabolites get perturbed simply because, as you can see, they're all interconnected, right? So, if I have an effect on one biochemical reaction, it's spread. Of course, the metabolites produced in that reaction are affected, but that effect also ripples across many other pathways, and you see changes of different kinds all across. And that's basically what this. So the point of this middle figure here is just simply to show you the metabolites are therefore linked in terms of, if you want to call it, expression levels and so on. And as a result, what you actually can see is you actually get a landscape of metabolites, right? And that's what the extreme panel shows. So you can map the topology of this landscape, and you can, you can compare it. Okay, and you can then look for changes in topology. Of course, that needs highly sophisticated analytical tools, which I'll just come to. And that's basically the principle of a strategy, is that can we map the topologies of the metabolome in an individual, the serum metabolome, and look for changes in those topologies that are characteristic of cancer. And to cut a long story short, this is finally what we arrived at. Obviously, the best and the most precise way to capture the metabolome or all the metabolites is by high precision mass spectrometry, so that we use the Thermo Fisher system because that very high resolution power. So we can actually capture a spectrum of between 5,000 to 8,000 metabolites in a given serum sample, right? So what that generates is, in a sense, a landscape representation like this in terms of the relative levels of metabolites that are present in the sample. And then we have actually generated a deep learning platform which consists of a series of artificial intelligence algorithms. There are a couple of algorithms for quality check because there are issues related to quality in terms of sample quality good, a technician getting careless or lazy, and so on. So we have a couple of algorithms that do quality checks, either support vector machines and a couple of others. Then there's another algorithm. Once the sample data, this is the mass spec data, it clears that, then there's an algorithm that kicks in and basically looks for whether there are signatures that correlate with cancer. So identify the sample as being cancer positive or negative. And in the event that it is positive, then there's another set of algorithms that kick in and analyze it, do a more deep dive into those signatures and tries to identify what type of cancer it is, the tissue for it. So that's basically a summation of our technology, and this is a kind of an illustration. This is the serum sample going into the mass spec, the landscape generated. We call this the software suite. Like I said, it's actually a series, it's a deep learning platform that integrates multiple AI algorithms in tandem, right? And so the first readout is it cancer negative or positive? And if it is cancer positive, the second readout that's generated is what type of cancer it likely is. So this is again a summary. I won't play this video. This is a video just actually describes the software. I'm just going to skip that. So basically the platform that we have developed is what we're calling OncoVerix. It's particularly important, detects all stages, but its USP is that it detects early stage cancers, as I'll show you in a minute. And all it requires is a very small amount of blood, so home collection, all of these are possible. It's a completely new strategy from what anybody else has tried before, particularly the AI algorithms and even the analytical strategy that we have followed. And obviously, since it's a screening platform, it's good for at-risk people and other people with genetic vulnerabilities and so on. And as the first step, when we were developing this technology, we first focused on the 4 women-specific cancers: breast, endometrial, cervical, and ovarian. I'm just summarizing the results here. You can see the sensitivity and specificity of over 5,000 samples tested. You can see the sensitivity and specificity that we got. It's really excellent. Specificity generally ranges between 98 to 100% depending on the test population. And tissue of origin, that means what type of cancer it is. And of course, we have patents, filed copyrights for software and so on. It's whatever ISO registered and so on. But this is what I wanted to emphasize. Like I said, our focus was on stage 1 cancers, and these are some of the results I'm showing you. These are actually published now. Okay, the age range is obviously the adult women, over 20 years to 70 plus, and you can see all of these are all samples from early stage, either stage 0 or stage 1, and you can see the sensitivity and specificity that we achieve it. And this is actually, we think, our unique USP which nobody else can offer in terms of early stage, because that is the critical stage for a screening tool, right? So this is something we're just bringing out in partnership with the diagnostic company as a laboratory developed product, right? We're just bringing it, testing the market, but this is really not the end product that we're aiming for. The end product that we're aiming for, since this technology seems to be so powerful and so sensitive, the idea is, can we actually incorporate more and more cancers into its detection spectrum? And the first stage, what we then did, tried to expand it to 15 cancers in women. These are the cancers. And you can see in terms of detecting, the sensitivities across the board are very high. The average sensitivity is about 98%. And in terms of identifying the tissue of origin of the cancer type, again, by and large, is quite high, and the average accuracy for tissue of identification is about 94%. And important here is this population comprises of both Caucasian and South Indian women. So there is at least some degree of ethnicity independence in how this works. Yeah. And subsequently, then, since that worked, then we went further ahead to cover both men and women, and we now have a test which covers 32 cancers across males and females, and we've just completed a field study across 22 hospitals in the country with about 5,000 samples, and these are the preliminary. I wouldn't take this too seriously. What this suggests is that it can work very nicely, and in a single test you can detect all of these cancers. And I'd like to just bring your attention back that most of these are extremely difficult to detect. There are no simple ways of detecting it, be it liver, kidney, brain, and CNS, and so on. So there we filed a provisional patent, but more importantly, to validate this and also get approval, we have now initiated a clinical trial with registered as a drug controller of India. This has just been initiated, and this is going to cover about 10,000 plus, a cohort size of 10,000 plus, and of course, whatever publication will do. But I think this is where we hope to come, and we hope to, if all goes well, we hope to bring it out by the middle of next year. Because the clinical trials are also already underway. So just wanted to show how powerful mass spectrometry is. If you get out of the box of the simple quantitation, targeted mass spectrometry and all, it's, particularly metabolomics, is a fabulous readout, is a fabulous snapshot of the entire health state of the body. And in principle, you can use this strategy for looking at any perturbation. Because remember, we map about 8,000 metabolites, and that is capturing the end point of the biochemistry of your body. And no matter what disease you talk about, these are always represent and are driven by perturbations in within. So I'll just stop there. This is just to show what I said earlier, that you can reduce mortality, and that's the team. We're about 20 people right now, and thank you very much.